currently who are here for our Nanoscale Microscopy 2025 final presentations here at the California Nanosystems Institute at UCLA. My name is Dr. Shailene Tanimoto, and I'm the Education Manager here at CNSI. And I've been with your students for the last week, watching them learn and grow, and we're very excited to showcase for you what they've learned over the last week. So first, I'd like to welcome you all to UCLA. UCLA was founded in 1919, and it's the second oldest school in the UC system. So our campus spans over 419 acres, which is a huge campus, and we offer over 330 undergraduate and graduate degree programs. We have over 33,000 undergraduate students. 7% of those are international students, 29% are first-generation students, and about 30% of those undergraduates are in the life and physical sciences. And then we are here today at the California Nanosystems Institute, and CNSI is one of Governor Gray Davis's Institutes for Science Innovation, and it was originally established in the year 2000 as an engine for innovation based on the belief that the defining technologies of the 21st century will arise from understanding and constructing functional nanosystems through controlled integration of nanoscale components. And so our mission here is to be a world-leading nexus of research, education, and translational activities by engaging in scientific discovery that fuels innovation and impacts society. So our education team here at CNSI, we are technically a department here at UCLA, but not a degree granting department. But instead, we're an interdisciplinary hub for research, technology, education, and entrepreneurship. And so our education team is led by Professor Sarah Tolbert, Dr. Rita Blake, our Director of Education, walking away right now, me, our Education Manager, and Dr. Elaine Marita, our Program Coordinator. And so we run various programs, including K-12 professional development workshops for teachers, a nanovation competition for 6th through 12th graders, public outreach events, and what you're all here for today are summer session programs for high school students. And so this program in particular is based on our CNSI technology centers. And so the CNSI has 6 open access technology centers that provide and develop leading edge equipment to users from around the world. And these are multi-million-dollar instruments that are very expensive to buy and maintain. And so CNSI aids in the purchase of these microscopes and has full-time staff who maintain and train users on these really amazing instruments. And so this week, your students had the privilege of learning about and using instruments in our microscopy centers here. And so you'll learn at UCLA we have a lot of really long, complicated acronyms, and CNSI is no different. And so these 3 technology centers, the Electron Imaging Center for Nanomachines, those are our electron microscopes. Then we have the Advanced Light Microscopy and Spectroscopy Lab. These are our light and fluorescence microscopes, and our Nano and Pico Characterization Lab, which are our scanning probe microscopes. And so this course, Nanoscale Microscopy, you can see the students had a very intense 5 days. And so this is a 1-week summer program for rising sophomore and graduating senior high school students from around the world. And this year we had 29 amazing students participate. So on the first day of the program, they were introduced to the basics of microscopy, and they built and modified their own microscope. Day 2, they learned about light and fluorescence microscopes. Day 3, they learned about electron microscopes. And day 4, they learned about scanning probe microscopes. And each afternoon, they had the opportunity to use these instruments in hands-on activity led by our technology center staff. And then on the first day, they were introduced to Journal Club, which is how to break down and read scientific articles. And they heard Journal Club presentations from our technology center staff. And then what you're going to hear from them today is how they worked in teams to dissect a research paper, which is what their presentations are on today. And so our learning objectives for this class are some skills in science, so particularly nanoscience at the Nanosystems Institute, but also about microscopy and about being a scientist. But we also know that not all of these students will go on to be scientists, so we have really important skills in life. So these are skills that are important to scientists, but are translatable to other applications in their life. So really important is their growth mindset, Your students had to work hard and persevere a lot through a lot of challenging material this week. But really important also is critical thinking, communication, collaboration, teamwork, empathy, creativity, curiosity, problem solving, and reflection and introspection. So we really want you guys to walk away with some of these skills and again, translate this to your whole life, particularly this critical thinking one. We need a lot more of that in our society. So a lot of amazing people helped to make this happen, and I'd just like to take a moment to thank some of them before we get to the presentations. So first, we have our nanoscale microscopy instructors, Dr. Laron Bentalila, Dr. Adam Stieg, Professor Hong Zhou, and Dr. Matthew Mecklenburg. These are people who run our technology centers every day, and they take the time out of their busy lives running those technology centers to teach your students. Next, we have our teaching assistants, our resident assistants, our lab assistants, and then all of the technology center staff who help to run the instruments for your students in those afternoon activities. So we'd like to give them a big thank you. And then lastly, we have our CNSI staff, audiovisual IT, digital communications, which is putting on our live stream right now, our events manager, Mitch, who helped to provide food for your students every day, and our work study students who helped to maintain our spaces. Okay, so now it's time for our final project presentations. So what we did differently this year is that we're doing this journal club presentation. So a journal club is kind of like a book club, but for scientists, where we meet and discuss published research articles to stay current with the latest finding and develop critical evaluation skills. So this provides a platform for evidence-based learning, encouraging participants to analyze research methods, results, and their implications. And so your students will present for 20 minutes on a portion of this article that we gave to everyone. And we'll have 3 minutes for questions from the audience. So I'm looking at you, audience. We need you to ask some questions. They should be able to explain this to someone who did not take this class and who does not know microscopy. So any question is a good question, and they should be able to answer those questions. So we highly encourage questions from family and friends. Otherwise, our amazing instructors are going to ask the harder questions. So again, parents, looking at you, ask some nice, easy questions for your students. I'm sure they would appreciate it. Okay, so this is our paper that we're talking about today, Microscopic Imaging Techniques for Molecular Assemblies, Electron, Atomic Force, and Confocal Microscopies. So this is a review paper that summarizes the common microscopy techniques that are used to study molecular assemblies, and it highlights a few applications of these techniques in specific research contexts. So the goal is that your students are explaining to you the microscopy technique, how it works, and then they'll talk a little bit about the applications of these. So this is our schedule for today. We'll have 3 groups present. Again, 20 minutes each, about 3 minutes for Q&A and like a couple minutes to transition them in and out. Then we'll take a 10-minute intermission around 11:20 to 11:30, and then we'll hear from the last 3 groups. So with that, I am under time. Nice, we're moving. Okay, so let's get group 1 up here to start.
Hello. Our names are Hunter, Alexander, Karika, and Yanming, and today we are presenting on elemental analysis using SEM, TEM, and STEM EDS. So in this presentation, here are our table of contents. We will begin by giving an explanation of these techniques, and then we will branch out into their functionality and results. So covering electron microscopy, imaging resolution, the difference between the different techniques, how electrons interact with samples, the principles of EDX, and applying the techniques. Electron microscopy, also known as EM, is a technique that uses a beam of electrons instead of light to image a sample. The electron beam is focused through magnetic lenses, as we can see here, onto the sample and then caught by a screen or a camera. The beam has an approximate wavelength of 0.0025 to 0.0038 nanometers, depending on the amount of energy, which is significantly smaller than visible light, which has a wavelength of 480 to 720 nanometers. Because of this, electron microscopy is used to image structures at the nanoscale, such as viruses or cell organelles. Samples must be examined in a vacuum chamber so that the electrons do not react with the air particles. In this paper, Table 1 compares the characteristics of different microscopy techniques, looking at these different criteria here. In this presentation, we'll just be focusing on TEM, SEM, and STEM, and our colleagues will cover the rest of them. The first characteristic that this table covers is imaging resolution, which is the ability to distinguish between closely spaced objects. A high resolution means that the image can differentiate very small particles. So electron microscopy has a very high imaging resolution because of the low wavelength of electrons. As we can see in this chart, it can differentiate significantly smaller particles than the human eye or a light microscope. It can even differentiate particles up to 1 nanometer. So what actually is 1 nanometer? Well, to understand how small it is, we can look at the smallest markings in a ruler, which are millimeters. Think that a nanometer is 1 million times smaller than a millimeter. So imagine 1 million particles spaced between these two ticks. That's how small the electron microscope can get. For sample preparation of the electron microscope techniques discussed, the subjects must have a stain on heavy metal such as uranium. This will protect the background from interacting with the electrons and will create an image where only the sample is shown. This is an example of an electron microscopy image, which was taken during this course, actually. And you can see that the background appears darker because that's where the stain is, and the lighter sample is shown because that's what's actually interacting with the electrons. However, everything has its advantages and disadvantages. In this case, while the high resolution is an advantage, it can only be performed on non-living samples, which is a disadvantage. This is because the vacuum, staining, and radiation will all kill the sample. Additionally, these techniques can be dangerous because the beam can damage the sample. The subject also must be dehydrated, which can change the shape and structure.
In the category of electron microscopy, there are 2 main branches: scanning electron microscopy, or SEM for short, and transmission electron microscopy, or TEM. SEM creates images by detecting secondary electrons that are scattered from the surface of the sample. This makes it ideal for an idea of a surface structure, giving us high quality resolution, almost 3D quality images. SEM has a broad field of view, operates at lower acceleration voltages, and requires minimal preparation for the sample, making it easy and reasonably quick for use. That being said, its resolution is sacrificed, typically about 0.5 nanometers, so while it is ideal for examination of textures and surface features, it can't quite reach atomic level resolution. TEM does this by recording electrons that have passed through an ultra-thin sample. This produces 2D projection images displaying internal detail of the sample at extremely high resolution. Because the electrons must travel through the material, this sample has a maximum thickness and is harder to prepare. Techniques like negative straining or cryofixation are typically required. Even with the increased complexity, the benefit of resolution is great. TEM can consistently achieve less than 50 picometers, allowing it to image structures such as viruses, protein complexes, or even single atoms. In our own imaging efforts throughout this program, we ourselves were amazed at how TEM images brought out detailed structural information that simply couldn't be seen in SEM scans. Here, Here we can see two images which were taken from electron microscopes during this program. As we can see, the TEM microscope shows much smaller particles. We can combine these techniques to create scanning transmission electron microscopy, or for short, STEM. Like TEM, STEM uses transmitted electrons, which means the electrons pass through the sample, and it also requires the sample to be very thin, typically less than 100 nanometers thick. But instead of lighting up the entire area at once, like how TEM does, STEM focuses a very fine electron beam on one small point at a time, and then scans across the sample in a grid pattern, similar to the way SEM works. This scanning process gives researchers more control over image contrast, and it allows them to collect several types of signals from a sample at once. These include bright field and dark field imaging, as well as X-ray spectroscopy, which can show the chemical elements present in different parts of the sample. One of the biggest advantages of STEM is that it keeps the extremely high resolution of TEM, often better than 50 picometers, but adds the ability to gather elemental and structural information at the same time. Because of these features, STEM is especially useful in materials science and nanotechnology, where scientists need to study not just what something looks like, but also what it's made of and how its atoms are arranged, all in one experiment.
Sorry about that. Let's start with the fundamentals. When we fire electrons at a sample, two key things happen: elastic scattering and inelastic scattering. First, elastic scattering is where electrons bounce off the atom nuclei like billiard balls on a pool table, giving us the structural information. And second, and more importantly, for EDS, inelastic scattering, where energy transfers create signals. The secondary electrons help us to see the surface topography. That's the standard SEM image. Backscattered electrons reveal composition and the contrast, where the heavier elements appear brighter. And then the real magic actually happens with the X-rays, which is the basis of the EDS elemental analysis. Here's how EDS works at the atomic level. The electron beam ejects an inner shell electron, for example, the K shell, exciting the atom, and then an outer shell electron fills the vacancy, releasing the energy. This is the energy that emits a characteristic X-ray, and its unique energy acts as a fingerprint for each of these elements. So we would ask which tool we should use for operating at lower voltages. SEM EDS uses about 5 to 30 kilovolts, and is perfect for bulky and thicker samples. And it gives us micron scale resolution, which is ideal for routine surface analysis. But when we need to see the atomic world, TEM and STEM ramp up to 300 kilovolts, requiring samples thinner than 100 nanometers. That's super thin. That's about 500 atoms that are stacked together, and in this extreme thinness, combined with fine electron probes, is what enables us with an unprecedented resolution. We're seeing individual atomic columns in advanced STEM, STM, sorry. And of course, none of these techniques are perfect. Yeah, we have common imaging issues like poor image quality, blurriness can be caused as seen in the figure, and pixelation or low resolution, and artifacts, noise, streaks, or distortions can be caused by equipment malfunctions, patient movement, or reconstruction errors, or the incorrect positioning can also cause the pictures to not be as detailed, and exposure issues like overexposure, too bright, or underexposure, too dark, and can be caused by incorrect settings in the X-ray or CT scanners. Blurring can also be caused due to the patient movement, and yeah.
Hello. So, since Parika already covered this slide, I'm just gonna give a brief overview. First of all, I want to ask, who here knows what an atom is? Okay, seems like everyone knows what an atom is. Essentially, an atom has atomic number, which differentiates it from other elements. And corresponding to this atomic number, there's a number of electrons for each element. And these varying number of electrons are in different positions for different elements. And using in energy dispersive X-ray spectroscopy, or EDX, you essentially bombard an atom with electrons. These electrons will then scatter with the electrons inside the atom, which will transfer some of its energy to the electron. The electron will be sent up a notch, or it will be given more energy. Then electrons don't like to be sent up a notch, so they would like to go back down. When they go back down, so the electron can be shot up like that, essentially. And when it relaxes back down, it emits something called the radiation energy, or in this case, it will be an X-ray. This X-ray is unique to each element, which is how you can detect it using a detector and differentiate the elements that you're seeing in a sample. So in this figure, we are given something called catalytic supermolecular hydrogel, which is just hydrogel. So these two structural formulas are the chemical representation of hydrogels. And this heme thingy is something that we're going to bind to these two hydrogels. In this image over here, we can see that it's an image created using a transmission electron microscope. So this image over here is the hydrogel without binding the heme onto the microscope. You don't really have to know what these compounds are, because they're just chemical. They're just so happen to be used in this research paper. And right next to it is the compound that is the hydrogel that is binded with heme. As you can see, it's a little thicker than the original picture, the original one without heme. The original ones are about 20 nanometers in thickness, and these ones are a little bigger. You can see from the sides. And the picture below is a picture with higher resolution, or which means you can see more clearly what in a closer up view. So pay attention to location E and location F over here. Location E is just a blank spot that you're going to place your grid in. This grid is made out of copper and is commonly used in electron microscopy. And using this technique called EDS that we mentioned earlier, you can see that copper is found, you can see a lot of copper here. Cu is the element symbol for copper. So using this method, you can see a lot of copper in this region. But there's no iron, which is an element in the heme, which part of the heme. So this is a picture of the heme-binded hydrogel, by the way. So in location F, it's closer to the structure itself, so you can see some iron actually next to the copper, which is the entire point of doing EDS here. Just want to see if the compound binds to the hydrogels. And this is another structure known as the semiconducting supermolecular block nanotubes. It's a long name. You don't really have to care what it is. It's just a tube, essentially, but nano. And this is an image captured by the STEM, the Scanning Transmission Electron Microscope. And using EDS, we could find out exactly where the iron and the copper, the carbon and the copper are. And we have highlighted them artificially using color. In this image over here, we highlighted the carbon atoms with orange and copper with green. If you look at the chemical formula over here, you can, nobody likes chemical formula, to be honest, but essentially, these 6 dots thingy, they're carbon. And at the end, you see carbon. No, at the end, you see copper somewhere. But yes, these nanotubes are they have the power to self-assemble. And they usually like to start out by making a they like to have the same element as when they start to form. So when they form, you can see a lot of carbon in the start, but then the copper, they're all concentrated at the end. This is due to the way it's formed. And using EDS, we can clearly see that this is how it forms. And this is just an image of the 2 pictures combined, essentially. Yeah, these are the works I did, and thank you. Does anyone have any questions?
I don't know, they're flashing at me. I was told red is bad, green is good. I don't know what it means when it flashes green and red. Now they're green. Okay. Jordan.
All right, thank you guys for your presentation. So can we go back to the last figure you just showed? I just want to clarify, so this coloring, what does that mean? Is there any physical significance to it?
They are artificially colored, so they're actually false colors, because using electron microscopy, you cannot identify the actual colors of elements, and nor do elements have their own color, actually, because they're super small and they don't emit light. They don't emit light, essentially.
I'll add on to that. It's to show the contrast and to show the carbon molecules and the copper molecules. So the red ones are shown the carbon molecules, and the green ones are shown as the carbon molecules. So like you can differentiate between the 2.
Okay, yeah, I think I see that. So as the electron beam interacts with your sample, it's interacting with the whole sample, but only X-rays for copper are emitted in a certain region, and for carbon, it's emitted in the same, but for copper, it's specific to one region, right?
Yes, exactly.
Okay, thank you.
Dr. Rita Blake.
So can you go back to slide 11, I think it is? You show electron microscope examples and you show like SEM versus TEM.
No. Or 11 is it
That
this
one, yeah. So you said it's, when you were talking about this slide, you said like it's clear that TEM is imaging smaller sample, or like you can see a better resolution, or you can see smaller things. But I don't know what we're looking at. There's sort of no context to the images that you're showing. So can you tell me like what we're actually looking at with the SEM and what we're looking at with the TEM, and what would be the scale of the things that we're looking at?
Yeah, sure. So both of these images were taken during this program. SEM was a mineral. I'm not really sure which type, but this was just one of them we were taking during the program. Then TEM, I think, is a type of enzyme. Yeah, that. So we just looked at both of these. And as you can see, like we can see clear in the mineral, but we can't see the molecules as well as we can see in that. So like TEM is just a lot more magnified than SEM is. It's a lot more closer up because we can see much smaller structures. I don't think these images have an exact scale, but I'm sure we could find that for you just looking at the equipment if like later.
And by using like the software we were shown, we can like look at how many nanometers exactly it was.
So you there was an earlier slide where you showed the different types of imaging techniques. Yeah, something similar to that table. Can you give an example of when you would use SEM versus when you would use TEM?
So you would like to use SEM when you're looking at the surface of an atom, because SEM is not SEM. You're using secondary electrons, which you don't usually go that deep into the sample. So you're just looking at the scattered back electrons, so you can really only see the surface of the atom. But TEM, you can the electrons are way more energetic, so they can actually travel into the sample, and you can see the inner structures of the atoms, or no, not the atoms, the cells.
For the TEM, you can look
Sample.
at the individual atomic columns, basically. So it would be like a more detailed image you would receive.
So, for example, if you want to look at, say, graphene, you want to look at the structure of graphene, you can just use SEM. You will see like somewhat hexagonal pattern, although it will be a little blurry because it's quite small. But TEM, you can look at cell organelles with TEM. You can look at each of the organelles inside the cell, essentially.
Yeah, or I think these photos illustrate it well. Like, if you want to look at a mineral and the layers in a mineral, then that would be a good thing to use SEM on. Or we looked at like hair, for example, and we could see it really up close, but we couldn't necessarily see the different molecules. So that was good for SEM. But if you want to look at molecules, then you should look at TEM because it has a higher resolution.
So I think a clarifying question here is like, you could look at the same sample in SEM and TEM, right? And what would you learn from SEM versus TEM, looking at the same sample?
First SEM, you would look at you would see the surface analysis, and for TEM, it's more like the atomic structure and like on a smaller level.
Right. So when you're talking about surface, you're talking about like a surface. We had a word for this, like a mountain range.
Topography.
Topography. Great. Yeah. And versus like a TEM, where you're transmitting the electrons through the sample, you're getting
Columns.
like when I talked about the light shining through, and then you get down here,
Image.
the 2D projection.
The 2D projection.
The 2D projection of that sample, right?
So just to clarify, the main difference between SEM and TEM is like where the camera is on it. So in TEM, the camera is at the bottom, which means that you're seeing the electrons that have gone through the sample, whereas at SEM, the camera is above and you're looking at the electrons that are scattered. So that's why it measures topography versus a 2D image. Does that make sense?
Great. All right. Thank you, Group 1. And thank you for our parent participation. We love that. All right, Group 2.
All right. Green means good. Green, green, green.
Good morning, everyone. Today, our group will introduce the exciting worlds of nanoscale imaging, focus on cryo email and TM. we will explore how these advanced techniques allow us to visualize molecular structures that are normally invisible to the naked eyes. Let's start with a simple question. How can we see the invisible? First, microscopy gives us the power to see objects far smaller than what our eyes can detect. At the nanoscale, these tools help scientists uncover the secrets of molecules and materials. Next, introduction to TM. In 1931, when Ernst Rusk and Maskell, they invented transmission electron microscopy, or TEM. This invention broke the resolution limits of traditional light microscopy. By using electron beams instead of light, TEM allows scientists to achieve nanometers and even sub-nanometers resolution. In 1980s, cryo-electron microscopies, or cryo-EM, was developed to solve TEM's major problems. Cryo-EM uses rapid freezing to preserve the simple nature shape, avoiding damage from drying. This method achieves near atomic resolution and has transformed the field of structural biology. In our presentation, we will talk about with four key sections. First, the difference between cryo-EM and TEM. Second, how cryo-EM works. Third, real-life research application. Fourth, our idea for future innovations.
What does cryo-EM mean? Cryo-EM is short for cryo-electron microscopy. Cryo means cold. In cryo-EM, samples are frozen very quickly at around -180 to -190 Celsius. This is typically done using liquid ethane cooled by liquid nitrogen. The goal is to create vitrified ice, a glass-like ice that avoids forming damaging crystals. Let's compare traditional TEM to cryo-EM. In traditional TEM, samples are dried and stained. Imagine a shriveled fruit. In cryo-EM, samples are flash frozen in water, like a fresh fruit. TEM causes more damage due to drying and heat, while cryo-EM minimizes damage by keeping samples cold. TEM works well for metals and hard materials, but cryo-EM is ideal for soft samples like proteins. Cryo-EM can also capture molecular snapshots over time, unlike TEM.
It's been a long time, since last time I do speech. I'm a little nervous right now. Never mind. Let's talk about how is the sample prepared. The figure on the right is standard single particle cryo electron microscopy workflow, and we are going to talk about how the sample prepared. First is the purified biomolecular are prepared, and cord is applied to an electron microscopy grid. The grid is glow discharged, so the carbon spot the film is hydrophilic, letting the aqueous sample spread. A drop of sample is placed on the grid, then blotted with filter paper to form a very thick liquid layer. The blotted and grid is plunged frozen by rapidly submerging it into liquid enzyme coated by liquid nitrogen. Because the remaining film is so thin, like under the 100 nanometer, and the cooling is so fast, on the order of like 10 to the power of 5 Celsius degree per second. The water in the film solidifies into a glassy ice instead of forming damaging ice crystal. The grid is then stored and transferred at cryogenic temperatures around 170 Celsius degree, which maintains the glassy state and locks the sample in place. So let's talk about why freezing works. Under these cryogenetic conditions, the frozen hydrate sample is imaged in the transition electron microscope. The glassy ice is transparent to electrons and preserves the molecule structures in a near native state. Because there are no larger ice crystals, there are no structure distortions or lattice diffraction. Instead, the water appears as featureless glass that surrounds the particles. The rapid freezing effectively fixes the biological assembly in its function conformation. Importantly, keeping the specimen at liquid nitrogen temperature also greatly slows the radiation damage in glass like ice. Molecules vibrate much more slowly and breakage by the electron beam is reduced together. These factors, vitrification of water and low temperature, also allow us to visualize biological structure which electron microscopy will persevering their native architecture. By following the procedure, electron microscopy enable high resolution imaging of proteins, viruses, and other assemblies with minimal artifacts. Rapid vitrification of the sample instead of chemical fixation or air drying keeps the molecular in place in their natural environment, making it possible to see the frozen hydrate structure under the TEM. The result is an image of the particles in amorphous ice closely reflecting their true and functional form.
Flash freezing acts like pressing a pause button on molecular motion. It instantly locks molecules in their exact position at a moment in time. This allows researchers to capture different steps in a dynamic process, like how a protein folds or how a fiber grows. Biological molecules are fragile and often collapse when dried. Cryo-EM avoids this by freezing the molecules in water, preserving their natural structure. The sample remains cold throughout the entire imaging process in the electron microscopy. Electrons are sent through the frozen samples to create high-resolution images.
So now I'm going to talk about the imaging resolution. So in cryo-EM, it typically achieves around 0.2 to 10 nanometers, which is very small. And this is close to and within the atomic level resolution. So it is particularly well suited for visualizing protein complexes and macromolecular assemblies in high detail. However, one limitation is that cryo-EM requires freezing the sample, so it's difficult to observe dynamic processes in real time. Additionally, cryo-EM is not typically used in in situ imaging, which is like imaging the sample in the process of doing something inside live cells or tissues. And the samples must be applied to thin film and vitrified, which restrict what you can analyze. But despite these challenges, cryo-EM remains a powerful tool for structural biology, especially when targeting static structures. In research, cryo-EM is widely used to uncover the structural basis of molecular self-assembly, especially in systems involving proteins, peptides, or synthetic biomolecules. And in the next 2 slides, I'm going to show 2 figures of 2 cases where cryo-EM is used effectively. So this is the first figure, and it highlights the power of cryo-EM as a snapshot imaging tool to study time-independent morphological changes in molecular assemblies. And because because these species or samples are frozen within seconds, cryo-EM allows researchers to preserve and visualize transient structures during dynamic processes that would otherwise be missed. Do you have the clicker? So in panel A, we see an example where cryo-EM was used to monitor the GTP induced structural changes in a tubular complex formed by PRD, dynamin, and PS. And dynamin is a GTPase that meditates vesicle fission during endocytosis. So in this first figure, you can see at 0 seconds before the GTP addition, the complex forms uniform tubular structures about 50 nanometers in diameter, and after GTP, progression of structural state appears. So at 5 seconds, cryo-EM reveals a mixture of non-constricted and constricted tubes, and by 210 seconds, most tubes are constricted to 40 nanometers, and dynamin-free liposomes appear. Notably, these intermediates were not visible with conventional TEM, highlighting the cryo-EM's advantage in minimizing sample preparation artifacts. In panels B and C, they show cryo-EM time course imaging of a nanofiber formation from FMOC ALA and FMOC HIS amylophilipic amino acid derivatives in the presence of Ag ions. And they use cryo-EM to visualize the kinetic stages of supramolecular polymerization, following with liquid to liquid-based separation, followed by nucleation and growth of nanofibers. This transition from solute-rich droplets to crystalline nanofibers demonstrates how cryo-EM can capture molecular scale phase transitions that drive self-assembly. So in the second figure, it demonstrates how cryo-EM in tomography and single particle cryo-EM have been extended in recent years to study artificial self-assemblies in small molecules, not just large biomolecular complexes. So in panel A, we see a chemical structure of a polyonic liquid which self-assembles into homopolymer nanoparticles smaller than 50 nanometers. And the 2D cryo-TEM image in panel B suggests an onion-like layered internal structure within 5 nanometers of spacing. However, the cryo 3D reconstruction from cryo-electron tomography in panel C reveals much more complex internal structure, going beyond what can be interpreted from 2D images, and it highlights how tomography adds a crucial depth to structure interpretation. In panel D, a protein-based artificial microtubule is using tetrametric soybean agglutinin and a rhodamine conjugated sugar. And using single particle cryo-EM, they resolve structure at a 7.9 nanometer resolution, confirming a well-ordered symmetric tubular assembly that mimics native cytoskeletal structures. And in panel E, a single-use particle cryo-EM studied a short 11 residue peptide DLIKGIS-VHI, which is a short peptide sequence composed of 11 amino acids derived from amyloid-forming TAR DNA-binding protein. And it was reconstructed at 3.7 nanometers resolution, revealing multiple distinct conformations within the same fibril, something that can't be captured by Volcker techniques. And these studies demonstrate that cryo-EM is no longer limited to just large protein complexes, and it now enables atomic and near atomic resolution insights into synthetic polymers, short peptides, and engineered nanosystems.
So I'm gonna talk about okay, so I'm gonna talk about the future of the cryo-EM, and this is some possible improvement of the microscope itself in the future. First is event-based direct electron detectors. So basically, when I did some research, now there's more like equipment like that, like Apollo, and basically it just shoot one electron at the time and give like smaller visual than the cryo-EM right now. And also some second is deep learning reconstruction stacks. So because when you look at all these kind of 3D models, they just don't come out like that because cryo-EM can only take 2D pictures at the time. So it's often constructed by like thousands of pictures and combined to create a 3D model like that. So in the future, I hope like there's gonna be like more advanced technique that can construct like 3D model faster than right now. And also, third point is continuous wave laser faceplates. It's basically now if you use cryo-EM, like many of the electron will be like lost, and like photon will be lost in the like background of the down to the 2D plate. But by having more advanced technique, hopefully we can get like more clear photos and more exact photos. Also, this is idea that I read like about how cryo-EM can have research in the future is like by cancer research. It's called ligand coated nanoparticles. Basically, it's for like future medicine. You put like a really tight like coat to the medicine, and like when it can like approach cancer cell and release the medical direct into the nucleus of the cancer cell and kill it. So now when you're trying to kill cancer, like often like you need like that like chemical or like radiations that will also damage a lot of healthy cells. So in the future, if this can come true, we can kill cancer cell more efficiently and like without harming other healthy cell in your bodies. And also, cryo-EM play an important role in virus because it can give you like a closed structure because cryo-EM can actually see through the virus, so it can give like a closer look, like and help scientists and researchers to develop vaccine towards the virus that they want to research on. And this is a picture on Nature, by the way. Like it's during the COVID-19 at the time, like they used cryo-EM to reconstruct like 3D models of the virus protein, I think. So in the future, if there's another pandemic, which I suppose there might be, and some days, hopefully not recently, scientists can use cryo-EM to figure out like the structure and figure out vaccine more faster than like usually in the past. And here's overall, this is for people like who's like sleeping during our presentation and like who's not paying attention. Just give you a overall like information that you can take away from our presentation. So basically, cryo-EM is a type of electron microscope, and the word cryo. Who know where the word cryo come from? Huh? Where it comes from? It's actually Yeah, it seems we've got a Greek expert here. It's from a Greek word means cold and frost. And also now the picture is just 2D that we need special software to construct a 3D models. And also, the most importantly, the cryo-EM allow people to observe biological sample because with EM, it conducts like high energy, it would just destroy all the living organism. And by frozen it, like it can release the electron beam, which like make them survive, and we can observe. And it can go about 0.122 nanometers, which is I think it's the smallest we can get using human technique. But it still releases a lot of energy, and it's radioactive. So you don't want to like break a cryo-EM like during research. And it's also really expensive. Thank you. Any questions?
Jordan.
Hi, thank you guys again. That was a really great presentation. If you go back, I think 5 or 6 slides, I want to look at one of the figures. I have a question. This one. So I see in figure C you have some like 3D kind of representation of some molecule, but in figure B I see these 2D representation. So are you saying that if you take a lot of 2D images with different orientations of the same molecule, you could reconstruct the 3D image of the molecule? So is that something you could do with cryo-EM?
Yeah, so if you take, let's just say a figure B picture, you have to take like a lot, like at least 1,000 pictures, and then like a human, or you can, in the future, you can use like more AI techniques, like to construct a 3D model that would result in something in like figure C.
All right, thank you.
Dr. Rita Blake. Again, parents, if you have questions, then students won't get hard questions.
So I have a sort of similar question. So in one slide you said it's difficult to evaluate the dynamic processes because of the freezing part of cryo, which is true. But I think in the slide before this, you showed that you can actually show dynamic processes in cryo. So if your whole sample is frozen, how do you actually get to see these dynamic, time-dependent processes?
Well, you'll need like a lot of samples.
Yeah, like basically if like you want to do a research using like any EM, you like at least it would take 5,000 of virus like to reconstruct one 3D models, like the one that I show, like the virus, and it's basically like 5,000 part of the picture combined together into a pictures.
I guess following up on Dr. Rita Blake's question, right? So in B and C here in these figures, in the top left part, they talk about initial stage, 3 minutes, 10 minutes, 3 hours. What does that time mean? Because these are time-dependent images of formation processes.
So basically, you can have like multiple samples. Like this is one. You take it 1 hour and put it under the cryo-EM, and the second one you can just like put it under cryo-EM after 2 hours.
So before you freeze it is how long that these have been sitting together.
Yeah, I think.
Correct. So you're still taking a snapshot and freezing the sample, but before you freeze it, you let the sample run a certain amount of time.
Yeah.
So then that allows us to get these dynamic processes, but we're still getting a snapshot in time when we take the cryo-EM image. Okay, great. Any other questions? Our audience, our student audience can also ask questions.
People are so quiet.
I'll ask a parent question, which means it'll be a dumb question, but just to relate this presentation with the last one. So in the last one, we're shooting electrons at the sample, and this one we're freezing it. Is it part of the microscope that actually does that process in both of them? Is it a component of a microscope, or do you do that separately?
First, there's no dumb question, just like what we said in the first, and thank you for your question. It's a process that off the microscope. So basically it depends on how cold you want to get. Like if you want to get like to like around I think 70K Kelvin, like you just put like liquid nitrogen on the top, and basically all the EM will like vacuum before, like because they don't electron to hit some like random thing and just explode the microscope, I guess. And like so they will like put liquid nitrogen in it, and which freeze the sample and like, but it's not putting it inside. It's just like around it. Inside's like vacuum. There's nothing like but the sample. And also, if you want to get colder, you can mix like helium, like liquid helium. I think it's helium-2 and helium-3, which can get to like 0.7 Kelvin, which is almost absolutely zero to get like any sample you want.
To get back to the crux of this question, could we go back to some of Kelly's slides on the sample preparation here? One more, I think. Go back one more. Yeah. Okay. So, both TEM and cryo-EM use the same physical instrument, an electron microscope, correct? But the difference between TEM and cryo-EM is here in this sample preparation. So the question was about, does the microscope do these different things, or is it in how you, the scientist, prepare the sample?
Yeah, we, the scientists, prepare the sample, and it's like it's a long time for preparation for the sample, and
this is much harder to do. Cryo is much harder to do to freeze these samples.
Often, like, cryo-EM can get to very small and can get living organism, but like this is a thing that why many researchers don't choose cryo-EM, because it takes a lot of time for the sample preparation, which I don't think our group mentioned a lot, because I think it's like more like we don't want people to think cryo-EM is bad, something like that.
Just takes a lot of skill.
Yeah.
All right, Jordan, how are we on time? We're over time. Okay, great. Thank you, guys. Okay, let's get group 3 up here.
Good morning, guys. Are we doing okay? Okay, so we will be going over in situ dynamics using liquid cell TEM. I'm Alan Lee.
I'm Anthony Mangayan.
I'm Felicia Lee.
I'm Daniela Lagos.
I'm Austin.
So in situ dynamics using liquid cell TEM, that may just sound like a bunch of like jargon, like words that you don't really know much of. So we will be going over what it is and how it works. This is our table of contents, what we will be going over, and I will be going into the principles. So what exactly is in situ dynamics using liquid cell TEM? So in situ is actually Latin for on site, or in its original place, its native state where it actually works, where processes happen. And then dynamics is like things as they happen in real time, events happening. So something is dynamically moving in your sample, right? And then liquid cell, which will be your liquid container that will hold your liquid for your sample. And then TEM, which is transmission electron microscopy, which are wonderful groups 1 and 2 kind of went over, but we will be briefly going over it again. So TEM is a powerful imaging technique that uses a beam of electrons to shoot at your super thin sample, and your contrast comes from your electron scattering. But with your scientific or engineering process, you have to identify a problem. And your problem right now is your vacuum environment will not allow you to see liquid samples inside liquid. And so they came up with a solution. Well, not yet. Here's the issue. It's in a vacuum. So like if, say, an astronaut is in space, and space is obviously a vacuum, right? They need a suit, or else if they just go out there, the liquids will like boil or like evaporate and it will collapse, right? So they need to have a suit. And your suit will be this special chamber, which is a microfluidic chamber, which means very small amounts of liquid sandwiched between two super thin electron transparent windows. And these are often made of silicon nitride, so your electrons can pass through and produce your image. And yeah. So we are also going to compare it to another TEM technique, which is environmental TEM, which is a variation that has gas inside the microscope, and it also allows in situ observations because it observes reactions that occur inside their original gas phases. And then, so we just wanted to compare it to liquid cell because it's pretty similar in seeing your sample inside its original state, where it's commonly used for looking at things as they grow, as they assemble, as they develop. And so you can look at your biological processes like cells and viruses as they do things. That's why it's dynamic.
Yeah, so moving on, we'll go into implementation and basically what's needed, and also the advantages and disadvantages of TEM and liquid cell TEM. So first of all, like just a very brief overview is that, TEM functions, you take an electron gun basically, and you have the sample and you shoot it down like this. And so the electrons, they'll pass through, and one common technique people use with electron microscopes is negative staining. So if you do photography, it's like a negative fill where it's like, you basically like invert like the blacks and the whites. So it's kind of the same here, because regular TEM is that like, here you can see like these, these images here. Regular TEM is that like, the dark is like your specimen, and light would be like background. Negative flips it, so your light. So here is like your light is your background. Your light is your subject, and your dark is your background here. And they do this by in the sample, like they'll put heavy metals in it, and so those heavy metals will scatter the electrons. So when the sensor tries to detect electrons, they'll be scattered and it'll end up dark. So the disadvantages of TEM in general is that it's in a vacuum. So obviously, you can't survive in a vacuum. You can try if you want to. Yeah, so nothing's going to live, so that's why liquid TEM was invented, or is going to be invented, yeah. And also, it's flat and monochrome, so yeah, it's 2D image. I know you can recreate it in 3D. It's kind of a pain, though. So the actual image itself is just going to be flat. It's going to be flat, 2D, black and white, all grayscale. All coloring is artificial. So this is like a bit hard to observe than fluorescence microscopy. Because that's all like natural colors, that's all legit. This is all faked. So also there's a risk of sample damage, albeit like it kind of depends on what you're doing. I know some microscopes they could go for like you could blast over 10 minutes straight until it starts to, like the cell starts to die. But like if you're doing like intense like imaging, or maybe you're doing a really high like powerful blast because in order to penetrate the sample, then you do risk damaging it due to ionizing radiation. So the advantage though is that it's really high res, which like compared to this is the main edge it has over all other forms of microscopy, is that like you could go literally to the atomic level. So like 0.1 nanometers of resolution, you see like individual atoms, see nucleic acids, see protein stuff, and also you see it has internal imaging. So you can see inside the cell, you can see like the individual organelles, like the nucleus, like Golgi apparatus, and so on and so on. It's like you're looking through a window down to a cell, if you go into a high enough resolution. And also we also have elemental mapping in these microscopes. So it basically works with the property that if you take an electron and you shoot it at a nucleus, it'll emit an X-ray. And so in the microscope there will be like an X-ray sensor to detect like these X-rays, and because each element has its own distinct wavelength it emits. Basically, you could like take it like I want to shoot electron here, and it'll say, oh, that's gold, and you keep doing that. So compared to liquid cell, obviously the really big advantage is that liquid cell has a living sample, because for biologists that stuff isn't very interesting. For chemists and physicists, I mean, they don't really look at living stuff a lot, so regular TEM works for them. And also you get to view these samples in water, so obviously cells don't just exist in a vacuum, they don't just like float around, they exist within liquids, so you can view them in their natural and living conditions. And also these changes are live, so unlike regular TEM where it's kind of like you're taking photos, here it's kind of like a live stream almost, so it's like as long as you just look into the microscope you could do whatever you want to the sample, you could view it in real time. And so moving on, we'll go into how to prepare the sample.
Okay, so I will talk about how samples are typically prepared. So liquid samples are placed in a small space between two thin, tough, and high transmittance membranes to create a sealed liquid cell. The cell contains the wet sample in a solution environment while still allowing the electron beam to pass through for imaging. So the sample has to be thin for the electron beam to penetrate and achieve optimal imaging conditions. So the liquid cell has two microchips with thin and electron-transparent membrane SIM windows, which create a microfluidic chamber to hold a liquid sample. A precise volume of liquid sample is placed onto the window of the bottom chip with an injector or by drop casting, which you can see in the image, ensuring a controlled environment with minimal evaporation and contamination, an even spread and no bubbles. The top and bottom chips are then aligned, squeezing the liquid sample. There are spacers on the chips that control the thickness of the liquid layer, which is typically within a few hundred nanometers. The chips are then sealed with O-rings, which prevent fluid or air from escaping as they act as a sealing device. Adhesives or mechanical pressure can also be used. This is done in an air-free or solvent-saturated atmosphere to prevent bubble formation and evaporation. Next, the liquid cell is mounted on a special TEM holder for imaging, paying attention to avoid air bubbles, overfilling, or damage. In Figure 5, you can see that the liquid sample is placed in a small space between two thin, tough, and high transmittance membranes, typically between 20 and 50 nanometers, completely enclosing the liquid cell. The liquid cell is a prepared sample environment that allows the electron beam to pass through for imaging while staying in a liquid state. The figure also shows the need for a thin liquid sample, with the sample staying in a narrow space between the two membranes. So here are some challenges. The membrane of the liquid cell acts as a barrier to isolate the liquid from the high vacuum. Isolation is crucial because high vacuum environments can cause liquids to evaporate quickly. However, the need for a liquid environment leads to many other challenges. The first challenge is the membrane, which must be thin, tough, and high transmittance to withstand the pressure difference and allow sufficient electron beam penetration and signal detection. Thicker membranes lead to increased electron scattering, which would degrade the resolution and contrast. There can also be electron beam damage, where the electron beam influences the sample's chemical structure. As the electron beam interacts with the sample, beam damage, or alteration or degradation of the sample can occur. The goal of liquid cell TEM is to observe dynamic processes over time, so continuous and repeated electron exposure increases the likelihood of damage. To check for beam effects, postmortem analysis like multi-IMS is done. Multi-IMS is a matrix-assisted laser desorption ionization imaging mass spectroscopy, in which the liquid cell is first separated into two membranes, and the sample on the membrane is dried before multi-IMS is performed and after a matrix is sprayed. The last challenge is resolution. Liquid cell TEM results in lower resolution than other techniques. The electron beam must pass through thicker material, and interaction and scattering of electrons within the liquid can degrade resolution.
Now I'll be going over what are the benefits of liquid cell TEM. To start, in comparison to conventional TEM or also cryo-EM, we are able to use liquid cell TEM to preserve the liquid state of the specimen inside a TEM vacuum and allows for in situ observation. LCTM or liquid cell TM allows us to understand the nanoparticle growth and assembly within the cell's natural state. We're able to use LCTM for providing high spatial resolution images, which are helpful when allowing researchers to visualize dynamics and processes in liquids with nanometer scale details. Moreover, we're able to use LCTM or liquid cell TM to reduce artifact introduction, as it's able to avoid using artifacts from traditional TM where the samples will be dried or freeze. This doesn't mean that the time it takes to create or prepare a sample is shorter. It only means the number of artifacts is reduced. Here we are able to see half of image 6. We are able to see in part H micelles fusion and solution. This is a time lapse of the fusion process of two micelles, something that we wouldn't be able to see in regular TM as the samples will be frozen. Similar to image H, image I also shows a different time lapse in fusion. This is the second part of figure 6. We are able to see in part M the LCTM images of the formation process of vesicles. And on part N, we're able to see a 30-frame average series of high spatial resolution images, something that we're only able to get from liquid cell TM instead of traditional TM, thanks to the high spatial resolution over a series of time. Moreover, how has liquid cell TM been used before? Liquid cell TM has been used in many other subjects like biology, in material sciences, chemical reactions, and nanomaterial research. In nanomaterial research, we're able to use LCTM to study nanoparticles in solution to see how they grow and interact. In biological studies, we're able to observe light cells to understand their behavior and process in real time. And for material sciences, we are able to examine how materials behave in different liquids, using this information to aid for new material development. And lastly, in chemical reactions, we're able to investigate how chemical reactions happen at the nanoscale level within a liquid. Furthermore, I would like to add a point, which is solid-liquid interfaces. This refers to the boundary where a solid material meets a liquid. And LCTM, we allow researchers are allowed to observe these interfaces in real time within their native liquid environments, enhancing our understanding of dynamic phenomena.
I'm gonna explain about the imaging resolution. The definition of the imaging resolution is the minimal distance between 2 points seen to separate. I used to think the two separate points gonna be like up and down. I don't know about yours, but it's actually the two responding points in a circle, such as the two closest highest point of the wave. And the image resolutions depend on the wavelengths. The short wavelengths improve high resolution because if you have lower resolution, such as the visible light, have 400 to 700 nanometers. If you give 100 sample in 400 nanometer, you will only observe it as once. And the TM has achieved below 0.1 nanometer, which is 4,000 times. About the LCTM, usually people think a longer title will improve the imaging resolution, but it actually sacrifices part of the imaging resolution to achieve the liquid cell functions. So, and I will show you why it's gonna decrease the image resolution. We can see from the images that there are two membrane and the liquid layer. When the electron beam gun shoots the electron beam through the layer, it will deflect and scatters. So it will be, since we know that the microscopy needs to focus, and the scatter will separate the channel and decrease the resolution. And there are actually four of them, but one of them is not scattered. It's very rarely, and the first one is thinner elastic scatter. It's like the shooting electron pass part of the energy to the electron of the specimen of the electron eat heat, and it will harm the sample. It will help the specimen for possible for the elastic scattering. It's like electron hit the electron. It's lose its direction and not in the correct path, and it will make your observation incorrect. And the last one, the ionization, it's like an upgrade of the inelastic scattering, because it's like if you the elastic beam shoot with high energy, it will release electron from what the atomic heat. And the last, there's actually four of them, and the last one is like about the higher charge electron. It's very rare, and it didn't happen a lot. It's all something about X-ray, and it didn't affect the results, so I didn't mention it here. And yeah,
Now I'll be going over some major points of our presentation. To start, liquid cell TEM or LCTM allows imaging of material liquids in a liquid environment at a nanoscale level. In situ observation allows real-time observation of dynamic processes like chemical reactions or biological interactions. LCTM is useful in materials such as science, biology, chemistry for studying structures and behaviors of samples. We are able to combine traditional electron microscopy or TEM with in situ studies to gather data in conditions similar to natural environments. And unlike traditional TEM, LCTM allows for the observation of living samples viewed live. And while you guys are able to look at our sources, I would like to add another point, which I think Group 2 went over, is the COVID-19 or the pandemic, and how we use the sciences to help us fight against the virus. I would like to add the LCTM, as you guys may be wondering, how does this matter to me? I'm not a science nurse or anything, it doesn't affect my life. Well, LCTM was actually a big part of fighting COVID-19 and creating vaccines, as we were able to understand how that virus worked to a nanoscale level. We were able to see how it moved around, how it interacted with other cells, and more. So that will be the end of our presentation. Thank you, and now we're open for any questions.
All right, questions, parent questions, yay.
Pardon my dumb question, but with all the energy going through the samples, how does it alter the dynamics of the living structure at all while you're taking the images?
Can you repeat that?
So when you're taking the images, I saw that there's a lot of energy going through the samples, and does that energy alter the living structure at all?
Yeah, so it actually does affect it a lot, but they're able to like control the dosage and amounts to make sure you get a good resolution without destroying it. So it does damage your sample, but you have to like balance and like control how much you do. Does that work?
You talked a lot about how to prepare the living cell sample. Is that preparation done here by individual lab members, or is that done at like a factory or like a commercial group that will do those kinds of preparations?
I think typically it's done within labs and within the own researchers, not usually in the factories or whatever.
So I feel like with this type of TEM, with like the range of motion that the specimen can move, there might be like specimen drift, or like they might move away from like the camera range. What are some ways to like prevent this, or how do researchers make sure that these specimens stay inside of that electron beam?
While I'm not sure how people are able to control the specimens, I do understand that the specimen is within a very small space, and while we're able to focus on one of them and follow it during a series of time, I believe the experiments are in way too long for them to just move around freely as much.
I had a question about figure 6, the second half of figure 6. So what are we looking at here, and why is liquid cell TEM really important and really the only way that you could probably visualize this?
So in the second part of figure 6, we're looking at vesicles and the process of formation of these vesicles. We can see this in part M, and on part N, we're able to see a better image or high spatial resolution.
So what is the vesicle? There's a key word here in this image because I read the paper.
I'm not sure of that.
Okay, so this is called a coacervate. Okay, so what is a coacervate? Sorry, I'm asking the hard questions. So a coacervate is a liquid-liquid phase separation, right? So you have one droplet of liquid inside another droplet of liquid, right? So the middle circle there is one liquid that is separated from the outside liquid. So I guess I answered my own question. This is why you need liquid cell, because these are two liquids, and if you used regular TEM, what would happen?
They will freeze together and you wouldn't be able to see the formation.
Yeah, you would dry out the sample, which means no more liquid, and so you wouldn't be able to distinguish the two liquids. Okay. Okay, one from Laurent.
Kind of following up on the question we had on the back here. First of all, I've really enjoyed your presentation. Thank you. And my question maybe is more like comments, and I don't know if you saw that in what you read, is to answer the question that was asked about drift, right? Maybe your sample is going to move. Maybe we can rephrase that and ask how fast can you acquire these images to get these time series? Do you have any idea of how fast you can image with a liquid cell?
So I believe you can view changes live through liquid cell TEM, if I'm correct.
I think you take like snapshots over time and like what are they called, like snapshots in time, and you can see how it changes like using different pictures.
Around 30 frames per second, if that makes any sense.
It makes total sense. Okay, thank you.
All right, thank you guys. All right, so that was our first 3 groups. So now we are going to take a brief intermission and get back here. Let's try to stick to our schedule. And so that would only be 2 minutes. Let's take a 5-minute break and be back here at 11:34.
good because they're the only ones that are doing it. No, the first three were about EM, and then there's two about ASM. And we're the only ones that are. No, so we're good because we're special. That's so true. That's what I'm saying. Oh my god, you two got go. Good. Bro, she's like 22. Specificity. Who's up next? Guys, group 4. All right. I know I saw it. Don't worry, I won't ask you too hard.
Okay, we are going to try to keep to our schedule. So we are going to get started again with group 4 coming on up. And while group 4 gets on up here, I'm going to give everyone a reminder about our microphone usage. So the tip that I gave all of our students in our practice this morning was pop star versus ice cream cone. If you hold the microphone like an ice cream cone, no one can hear you. They can hear me because I'm loud, okay? But you want to hold it like a pop star. So that means that if you turn your head to look at the screen and you keep talking and the microphone didn't go with you, they won't be able to hear you, okay? So when you turn your head, the microphone goes with you like you're a little pop star, okay? So we're all going to channel our inner Sabrina Carpenter. Adam, do you get this reference? Yes? Nice. And we're going to please, please, please hold the microphone correctly. I'm hip and cool, guys. Okay, come on, group 4. All right. Green, 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 green. That's on. Hello?
You can prop it on the chair if it's too, like in front.
Okay, good morning everyone. Today we'll be talking about mechanical properties measurement using atomic force microscopy, AFM for short.
I'm Adrienne.
I'm Brianna Huang.
I'm Caroline Huang.
I'm Timothy Lentz.
And I'm Yellen Bian. So this is an overview of our presentation today. We'll be talking about general AFM working properties, force measurement using an AFM probe, how the samples are prepared for force measurements, which mechanical properties can be determined by AFM, what nano indentation is and its applications, and the imaging resolutions produced by AFM. So to start off, what is atomic force microscopy? AFM is an imaging technique that detects atomic force between the tip of the microscope and the sample surface. As a tip makes contact with the surface of the sample, the cantilever bends, and the bending is detected using a laser diode and a split photodetector. So this is the cantilever. A cantilever is a beam that is secured on one side and can be moved depending on the tip. And the laser is shown down into the cantilever and reflected off into the photodiode. A photodiode is a sensor that records the movement of the lasers. If you see on the GIF over here, this is how it functions. As the tip moves, depending on the surface, the laser also moves with it, and the laser's movement is recorded on the photodetector. The photodetector detects two things. Thank you. The photodetector detects two things: deflection and topography. Deflection is the bending of the cantilever, while the topography is the surface level of each sample. So there are three main working properties in AFM: force measurement, topographic imaging, and manipulation. Force measurement is the measurement of forces between the probe tip and the sample surface. Topographic imaging provides high-resolution 3D images of a sample's surface topography. These two pictures down here are great examples of topographic imaging. If you look over here, the darker colors represent the deeper levels, while the lighter colors represent the higher levels. Lastly, manipulation is how AFM can be used to physically manipulate surfaces at the nanoscale by controlling the forces between the tip and the sample.
Thinking of the AFM cantilever as a mini divine board, we know it's bringing constant K from calibration. Whenever the tip pushes or pulls, the board bends by distance delta. Huxell tells us force is simply F equal to K times distance. A tiny laser dot reflects off the back of that board onto a split photodiode. Two light sensors side by side. If the board bends, the dot slips towards one half. Electronics translate the slip into a voltage, and software turns the voltage into a bend measure in nanometers. A piezo stage now moves the sample up and down. During approach and pullback, the system logs force versus distance, giving a force-distance curve. From that single curve, we pick out the first touch point, the adhesion dip, and the contact slope that rests faster on hard materials. Instead of one curve, the AFM repeats the process every time pixel in a scan, tens of thousands of times. Each contact slope is converted to a local Young's modulus. Put all those values together and you get a color map that shows soft and hard zones across the surface.
Okay, so how are samples typically prepared for force measurement? Number one, we have substrate selection. The most commonly used substrate for AFM is called mica. Mica is a naturally occurring mineral that can be easily cleaved to create a smooth, atomically flat surface. This flat surface allows for the imaging of various molecules and materials, especially biological samples. Second, we have sample mounting. Adhesion ensures samples is strongly adhered to the substrate in order to prevent it from being pushed around by the AFM tip. Vibration and noise can interfere with the measurements. Sample mounting is crucial to minimize distracting factors. Solvent removal ensures that the measurement won't be interfered during experiments. Number three, there are specific sample types. Particulate samples often deposited onto a substrate, dried and potentially rinsed to remove excess material. DNA and RNA can be prepared by dispersing in a solution, then deposited onto a substrate and dried.
So first of all, what is a mechanical property? It's a characteristic of a material that describes its behavior under applied forces or loads and can indicate how a material will react under stress, strain, and other mechanical influences.
Some key mechanical properties include strength, which includes tensile strength, yield strength, and compressive strength, as well as ductility, malleability, hardness, toughness, brittleness, and stiffness. And the AFM would theoretically be able to characterize all of these, but would likely need to know more parameters about the sample, such as the composition of the material. You can also use the Young's modulus data to calculate some of these, which will be explained later on. Now, rather than just general mechanical properties, what are most common for the AFM to characterize? Some of these include the elastic modulus, or stiffness, which is also the Young's modulus, as well as viscoelastic moduli, adhesion, and friction. Elastic modulus, or stiffness, refers to the resistance to elastic or non-permanent deformation under stress. An example of this would be steel having a high elastic modulus, while rubber has a low elastic modulus due to how it's easily deformed. Viscoelastic moduli refers to modulus depending on the frequency at which it's being measured. And an example of this would be silly putty or oobleck, in which the stiffness depends on how fast you pull it and how hard you hit it. Adhesion refers to the sample surface having an attractive force, which can interact with the probe tip. An example of this would be the water droplets sticking on top of a leaf. And finally, friction, which is a force that resists motion between surfaces in contact. And an example of this would be when you rub your hands together. These are parts of Figure 9. And as you can see from Figure 9b, which is right over there, the AFM helped us visualize the nanotubes, and through that, researchers figured out the diameter of the nanotubes, which is 80 to 300 nanometers. Figure C shows a histogram of the nanotubes, right over there, and allowed the researchers to visualize the distribution of data regarding stiffness. Through this, they found that the average Young's modulus of the nanotubes was 19 gigapascals. Next is Figure 9F, which is a composition of the Young's modulus of the materials. And through the paper, we learned that the prolyl diphenylalanine assembled to form a helical structures, unlike diphenylalanine. Figure 9F shows that the helix-like peptide formation resulted in one of the highest Young's modulus, or the greatest stiffness, of the biological material, and showed us that the AFM can help researchers compare the characteristics of different materials.
So what is nanoindentation? Nanoindentation is a specific AFM technique that is used to measure the mechanical properties of your sample. You pretty much take the AFM and you press the probe onto the surface of your sample to feel around, and the force is measured by tracking how much the cantilever bends, and then you can also measure indentation depth, which is how far the tip goes into the material without breaking the sample. This data is used to create force distance curves, which is this one. The blue line is when the probe approaches the sample, and the red is when you're moving the probe away from the sample. These force distance curves tell us about the Young's modulus, which, as you know, is the stiffness, and if the material deformation is elastic, which is temporary, or plastic, permanent. As you can see on this image, this material deformation is plastic, how it's keeping it's deformed. And then here in Figure A, you can see an example force distance curve where at point 1, the probe is away from the sample, so there's no force being applied. At point 2, that's when the probe starts to contact the sample, and then that measures the Young's modulus, or the stiffness. And then probe 3 is when you start retracting the probe away from the sample, and that measures the adhesion. That's where you see the negative force here. And then on 4 and 5, it's also adhesion, but it's either non-specific interaction or a specific interaction. A non-specific interaction would be like a weak physical force, like van der Waals, between the atoms. And then a specific interaction would be specific to your sample. So say like your sample has binding receptors that attach to the probe. And then nano-indentation in use. So this is also from Figure 9, as we saw earlier. The D here is the pro-Fifi molecule. And we use nano-indentation to measure the Young's modulus of it. So pro-Fifi is a tripeptide that assembles into a helical structure. And we measured a Young's modulus of 44 gigapascals, and this is a lot higher than average biological materials. So you can see how this helical structure gives it like a lot of support and stiffness. And then we also have another one, which is HypeFiFi, which is another version of D, but it has a hydroxyl group, so there's more hydrogen bonds. And then after measuring the Young's modulus, it has a Young's modulus of about 108 gigapascals. So you can see how the hydrogen bonding makes the molecule a lot stiffer because it's bonded together more firmly. And then AFM imaging resolution. The imaging resolution is very dependent on how sharp the tip is. Usually these tips are ideally one atom at the bottom. It can usually be a little off, but mostly it's just a couple atoms wide. And then for AFM, we have two main types of imaging resolution. We have vertical resolution, which is the z-axis, and then lateral resolution, which is just the flat 2D image. So lateral resolution is mainly between 5 to 15 nanometers, and then vertical resolution is a lot more precise. It can get down to angstrom level, which is fractions of nanometers.
For figure 14, researchers posted an photopolymers one hydrogel layer, then repeat to add a second layer on top. They deliberately make the bottom layer stiff and the top layer soft. And for B, shows two things. First is a confocal laser image where different fluorescent dyes prove the two layers are in the right place. And the second is an AFM mechanical map. This map is built exactly the way I just described, 1 force distance curve per pixel. Darker colors mark the soft top layer, and the brighter colors mark the stiff base. So because of the AFM converts cantilever bends into force and then force into modulus, it gives solid quantitative proof that fabrication worked.
So part C and D of figure 14 focuses on AFM mechanical mapping of the two different hydrogels named hydrogel 1 and hydrogel 4. Hydrogel 1 on the left has a lower stiffness range, while hydrogel 4 on the right has a much higher stiffness. These images shows how stiff different regions of the hydrogel are spatially resolved. On the top right, these graphs compares force deformation curves for hydrogel 1 and 4. According to the graph, hydrogel 4 has resists deformation more, confirming its higher stiffness. Part D reinforces that hydrogel 4 is over 3 times stiffer than hydrogel 1. The graph on the right shows a transition across a spatial region. Moving from hydrogel 4 to 1 shows a sharp drop in stiffness, illustrating a well-defined interface between the two hydrogels. The one at the very bottom shows a transition from high stiffness, which is hydrogel 4, to low stiffness, hydrogel 1.
This is a summary of everything we talked about today. First up is what AFM is, which is a microscopy technique that uses a cantilever system and an atomically sized probe to achieve high resolutions. An AFM measures force by detecting the cantilever deflection with a laser system and converting that deflection using Hooke's law. Then samples are typically fixed on a clean and flat surface to ensure stability and accuracy during AFM force measurements. Some mechanical properties that an AFM can characterize includes Young's modulus, viscoelastic moduli, adhesion, and friction. Nanoindentation is useful when measuring mechanical properties of samples, such as Young's modulus and adhesion strength. And finally, the imaging resolution of AFM is dependent on how sharp the probe is, but generally images can have a resolution of 5 to 15 nanometers laterally and fractions of nanometers vertically. These are our citations. Any questions?
Okay, Jordan.
Hi guys, fantastic job. So I just wanted to re-clarify with AFM, there's no like transmission or anything regarding like particles hitting a detector, right? This is like a physical object interacting with the sample. Am I correct in that assumption?
Yes.
So it's like using, instead of using our eyes, we're using like our hands to create a 3D object, right?
That's correct.
Cool, thank you.
Dr. Rita Blake.
Can you go back to the image of figure 14A? Do you know for this image, like I don't know if you had a chance to go back and read the original paper where this is from. I'm trying to understand this interface layer in the middle. Is that like some kind of other material to help adhere the two, like the first and the second layer? Does it have any different properties from either of those layers? And how thick is it relative to those layers? Because the image here makes it seem like there's like some significance to it when you zoom in, but then in the non-zoomed in photo, it seems like it's hardly there. So I'm just curious if you got to read the paper a little bit and know sort of what that interface is and what it's doing.
Well, I mean, that's a good question. That's kind of like out of my knowledge, so.
We can infer that it might be a layer to connect the first and second layer, since without an interface they would move around, but we're not really sure about the specifics of it.
But when you do nano indentation, wouldn't that damage the probe too?
Yeah, it kind of depends on how much force you're applying onto the sample. If you use too much force, it can either break the probe and the sample. So you kind of want to balance it out so that you can get a good reading of data without damaging your sample.
So you have a loop controlling what's happening, right?
Yeah.
And also the tip is not sharp, it's more like rounded. And so when it moves, it won't like stab your sample.
Okay, thanks guys for the presentation. Quick question, can you go to the next slide? So nano indentation can be done without a microscope. Right, so we have, next to where you were sitting yesterday, there's a machine called a rheometer that does bulk mechanical measurements. There are nano indenters that just poke at things. And so I'm wondering if you can just comment on why we might be combining this with a microscope and why you'd be interested in getting spatially resolved mechanical measurements instead of just a measurement on the whole material. And the answer is in your image here. But I'm wondering if you thought about why do we want to do it with a microscope instead of just a bulk measurement?
I think we can infer that using a microscope, we would be able to get a lot more different measurements. So we would get a lot more information about the material, and that can help us with further research experiments.
So you can see in C that it's not the same everywhere, right? You have spatial variation in your mechanical properties, and also in your histograms, there's not one value. There's a distribution of values, right? And so that's where the microscopy side of things unlocks additional information. But yes, you're right. Thanks.
All right, thank you guys. Okay, group 5.
Hello? Hi,
So today we're going to be talking about high-speed and environmental AFN for imaging molecular dynamics. I'm Mia.
I'm Shayna Masudi.
I'm Sophia Loringer.
I'm Jake Shin.
I'm Ben Scott.
And now we'll begin. Okay, so this is our table of contents. First, we're going to...
Sorry.
Okay. First, we're going to go over an introduction into high-speed AFM. Then we're going to talk about scanning biological materials. Then we're going to talk about in situ AFM, applications, environmental AFM, and then we will conclude. So, first I'm going to give you a background on what conventional AFM is, which the group before also touched upon, but I'm just going to talk about it again. So AFM, also known as atomic force microscopy, was invented in 1986 by 3 IBM scientists named, and they're in the corner over there, named Gerd Binnig, Christoph Gerber, and Calvin Quate. They also invented the scanning tunneling microscope a couple years prior. However, that microscope could only image samples that were conductors or semiconductors, which made the field a little bit limited. And then they invented the AFM, which made it possible to image a lot of other surfaces, including insulators, biological specimens, polymers, and more. So taking a step back, AFM is a form of scanning probe microscopy, which is the atomic force between the tip and sample surface. Structurally, the AFM uses a cantilever, which is right here, and that's sort of like a little diving board, and at the end right there is the tip. And so the tip apex, which is like the very end of the tip, influence how the probe interacts and interprets the sample. The sharpness of the tip, similar to a numerical aperture, which is like the light gathering ability of the object, is inversely proportional to the resolution of the image. And resolution is the smallest distance between 2 points on a specimen, being distinguishable as separate entities. And so that means like as you zoom in closer and closer into a photo, once you can no longer establish the objects separately and they all start blurring together, that's when you start lowering your resolution. And so now I'm going to talk more about high-speed AFM, Okay, so high-speed AFM is similar to AFM in terms of the basic principles. However, like in the name, high speed, it works significantly faster than AFM. This means that it requires a lower imaging force, which exerts a minimal force from the tip onto the sample. So referring to this graphic over here, the excitation piezoelectric makes the cantilever, which is the diving board thing, vibrate really fast so that it can effectively scan the surface of the sample. And then the photodetector detects how the cantilever moves, measuring the position of the laser, and then the laser shines a beam onto the cantilever to track the movement. So high-speed AFM uses a thinner cantilever than conventional AFM, and that's because it's more lightweight, so you're able to go through the image a lot faster to capture all of the images at a really rapid pace. It also has a high spatial and temporal resolution so that you can monitor the dynamics of a variety of specimens, including supramolecular polymers. It is AFM that allows the simultaneous assessment of structures and dynamics like in real time. So like I showed in the top corner, it's like watching a movie at the molecular level. You get to see everything happen as like in real time. And so I thought about it like in colloquial terms, HSAFM is like AFM on steroids, although it has some differences, but that's like an easier way to look at it. And additionally, HSAFM allows imaging of wet specimens with nanometer resolution, capturing the fast motion of native proteins at the molecular level. And like all kinds of scientific measuring, it does have its drawbacks. In this scenario, there is a higher rate of damage possible to the sample because of how fast you're moving. There's also like the spatial resolution is reduced, again, because of how fast you're moving, and it's more costly than the AFM. But it has made a lot of contributions to science and is really helpful in measuring samples, which Sophia is going to touch upon right now.
Thank you, Mia. Okay, so high-speed AFM allows scientists to observe these biological materials and processes by capturing rapid images, and we can see frame-by-frame images of how molecules move, grow, and interact. So, for example, we have figure 16 from the journal. which is basically examples of high-speed AFM images. So in panel A, we see actin. Let's see, this right here. This is actin, which is a microscopic protein inside of our cells that help them move and divide and keep their shape. So using high-speed AFM, we've captured several images of actin from 0 to 13.5 seconds. And essentially from 0 to 2.5 seconds, the scientists have used this AFM probe to apply pressure on the actin. And you can see in the gap between, in around 2.5 seconds right here is where the pressure is being applied. And you can see the scientists were able to change the ends of the actin and move the filament away from each other. So this happens very quickly. 0 to 13.5 seconds is really fast for a reaction like this. And with normal AFM, we're not able to see processes like this, where normal AFM, it would be anywhere from minutes to hours to create a high-resolution image. So high-speed AFM lets us see things like this much quicker. Moving on, in panels B, D, and C, we're looking at amyloid beta protein, which is found in the brain. And when this protein clumps together, it's known to form fibrils, which are tangled strands, and are eventually known to cause things like Alzheimer's. And in the panels, these are called chymographs. So it's basically a compilation of snapshots, like a time lapse, but you're seeing a frame by frame condensed into one image. So imagine you're looking at a thermostat and you're in a room and it's heating up, and you take an image of the thermostat as the temperature increases and you condense it into one photo. So you can see how like the temperature is like increasing, it looks like a slope. That's kind of what we're looking at with the chymographs right here. So each of these like little lines are a different image taken by the high-speed AFM of the amyloid beta protein. And in AFM, basically when the probe is scanning the surface, as it moves higher up, the surface is going to be taller, and the contrast is going to be lighter. So we can see the lighter sections right here are going to be higher up, and that means the protein is more coiled. It's more spiral because there's more material. So with this chymograph and the high-speed AFM, we could see the spiral shape. This is the spiral type up here, which means more coiled, that's lighter colored. And then down here we can see the straight kind, and then this is hybrid, where it's a combination of the more flat and the higher. And the spiral fibrils spread more easily, which can speed up the progression of the disease. And the straight fibrils can accumulate and persist longer in the brain, which can cause long-term damage. And the hybrid can be highly toxic and resistant to treatment, which is especially dangerous. So because high-speed AFM can scan things like this so quickly, like I think this is over about 26 minutes is we're seeing this process. We're able to see this in real time, and we can see how it's growing and changing over time faster, which traditional AFM, you would need a lot longer to scan each sample.
So similar to Sophia's panels A, B, C, and D, panels E, F, and G are also high-speed AFM images, but we're looking at the FTSK protein. And so in panel E, it's an image that shows DNA strands lying on the surface, these yellow and orange strands right there, with little white circles, which are the FTSK proteins attached to them. So through this image, we can see that some are spiral looped and straight. And this tells us that DNA takes on different shapes and that these shapes may affect how the proteins interact with them. The key takeaway from this panel is that the protein FTSK binds to DNA in different ways depending on the shape of the DNA. And in panel F, it's a bar graph that shows how often the FTSK protein interacts with each shape of DNA. So we can see that the spiral DNA has the most interactions with the FTSK protein, while it happens less on the straight and medium with the hybrid. And the key takeaway for this panel is that the FTSK protein seems to prefer spiral-shaped DNA in comparison to the straight and hybrid. And in panel G, it's a bar graph that shows how fast the FTSK protein moves along the DNA. And so in this one, we're comparing spiral and straight, and we can see the FTSK protein moves faster along the spiral-shaped DNA compared to the straight DNA. So through this, we can see that the shape of DNA affects how fast the FTSK protein interacts with DNA. And this matters because it shows that the shape of DNA changes how proteins behave. And that's important because proteins like FTSK are involved in DNA repair, packaging, and copying, which are essential for life. And with high-speed AFM, we're able to see this happen in real time, and at the nanometer scale, something no regular microscope can do.
So I'm going to be talking about in situ AFM. I know it was kind of touched with another one of the groups with transmission electron microscopy, but you can also use it for atomic force microscopy. So what is in situ AFM? So in situ atomic force microscopy is the use of external stimulus during regular AFM to cause the sample/specimen to react as you're scanning it. So what does that mean? It's basically saying I can apply any external stimulus and that will cause the sample to change. And because we're using high-speed atomic force microscopy, we can see how the actual sample reacts. Also, there's very little preparation for in situ AFM. It's able to, that's what's great about it, it's able to show exactly how cells and different proteins, different biological structures would react normally in the body. So different external stimuli you can use would be like temperature, magnetic fields, physically touching the specimen with the probe, and then applying voltage. So here's a couple different ways that you would use in situ within biology. So with figure A, you can see EAK 16-2 fibers, which they're basically just self-assembling proteins. So you can see how it has like a hydrophilic side, a hydrophobic side, and then we place that onto a mica surface. So I know that was kind of touched upon, but why you would use mica surface, it's chemically inert, it's really easy, which means it won't react. It's really easy to make it smooth, and it's easily cleaved. Also, another thing is it's naturally has a negative charge, so we can have the sample, the protein, interact with the mica surface and stay stable. In figures C and D, you can kind of see it's kind of hard, but there's a small little fragment that has broken off by using the atomic force microscope probe. So we're using mechanical force to force in E and F. You can see that it's slowly growing back, and we can see that using high-speed AFM. Finally, in here we're using peptoids in this next figure. So peptoids are just like peptides, they are just have a modified backbone. So in H, I, and J, you can see them actually self-assemble and start to imitate what is called a lipid bilayer, which is what a cell membrane is made of. And then in K, L, and M, you can see cracks, which are mechanical force applied by the AFM probes. And then they slowly are healed over time. So just showing how we're utilizing high-speed AFM to see how these different proteins are responding to that stimulus and regrowing.
Thank you. So I'm going to be discussing applications of high-speed AFM using figure 17 as an example. So in section A of figure 17 that you can see over here, it's showing two derivatives of porphyrin, porphyrin 1 and porphyrin 2. And then moving on to section B, it shows that porphyrin 1 can self-assemble into a stable nanofiber structure. But before it becomes a stable nanofiber structure, it gets stuck in a kinetically trapped nanoparticle form, which is porphyrin 2. And kinetically trapped means that it's less stable. It has trouble transforming into this final nanofiber structure, and it is quicker to form. So moving on to section C, you can see images of a nanofiber structure. And what scientists did was they took preformed nanofibers called seeds, and they placed it onto a smooth, flat material for imaging.
And what they did next was they took the porphyrin 2, the nanoparticles, and they added it to the surface. And what happened was that the nanoparticles assembled onto the nanofibers, and that's why you can see elongation in these images taken over time. So what high-speed AFM allowed, and this is called seeded polymerization, this process. So what high-speed AFM allowed scientists to do was take images every 0.1 second. So they were able to watch this whole process in real time. There, as you can see, there's 2.4 seconds, 2.7 seconds. And if you look at the last image at 5.8 seconds, you see that the nanofiber suddenly is about half the size of the image before. And this is due to breakage of the nanofiber, but it's most likely due to the nanofiber coming in contact with the probe and the force that results from that. So what scientists found was that the average growth rate was 12.4 nanometers per second, or 35 porphyrin molecules added per second. So what we can take away from this figure specifically is that high-speed AFM allows us to observe and even measure molecular self-assembly. And it takes images so quick that it's kind of like a flip book. Like, you know, when you like flip through the pictures, it kind of creates a movie. This is kind of the same thing, where you're getting so many images over time that you get to watch the whole thing from beginning to end. So next, I want to go into environmental AFM, which is the use of an atomic force microscope to study materials and biological samples in controlled environmental conditions. So this could mean in liquid, in gas, in different temperatures. You can make it hot, you can make it cold. And the point of this is to examine the behavior of a sample under specific environmental conditions, and this would usually mean the natural environment of the sample. So, for example, hydrogels. Hydrogels are used to grow tissues, and it's kind of in the name, hydro means water. So hydrogels absorb a lot of water, so you would be able to keep the hydrogel in water while doing imaging, so you can get kind of more accurate real-life results. And back to Jake.
So before we go more into imaging resolution, I think it's important to understand what that is. So imaging resolution is basically how you can tell two things apart, and the better the resolution, the smaller the things you can tell apart. So example would be you're walking down the sidewalk, you see some ants walking by, and if you're far away, the ants just look like black dots, but if you get closer, maybe you can see some legs, some arms. But if you get a really powerful microscope or magnifying glass, you can see their eyes or whatever they're carrying. And that's basically what resolution does. It lets us see finer and finer details. And with high-speed atomic force microscopy, we're looking at things a million times smaller than ants, like individual molecules. And not just still images, we're watching them move, fold, and interact in real time. High-speed AFM is able to produce images at the resolution of less than or equal to 1 to 50 nanometers. So things like viruses, DNA, and protein, as seen on figure 16 and 17. So this is another application in which high-speed AFM can be used in the real world. It's used to visualize how insulin molecules break apart at the nanoscale in real time. So scientists could actually see how and when insulin molecules begin to stick together. And using this insight, researchers were able to design modified versions of insulin, like insulin lispro or insulin aspart, that are less likely to clump. And when they clump, the insulin pens, they tend not to work as much. So these new versions stay in their active form longer and work faster once injected. And so through high-speed AFM, we're able to understand how insulin behaves in pens and pumps over time. And this led to more stable insulin formulas that don't break down as easily, which is important for people using insulin pumps or storing insulin long term.
Thank you for listening, and now we're going to open the floor to questions.
All right, student questions.
Okay, so you said the high AFM takes about 26 minutes, right? How long would a regular AFM take?
I don't know if you remember, but yesterday we saw one of the images, which was like super detailed, and it took them 16 hours to image that image. So it can take from like anywhere to like minutes to hours, but high-speed AFM can actually work in seconds, like I showed in the figure where it was like 0 to 13 seconds. They took like 6 images, so it works much faster than normal AFM.
How does the piezoelectric excitation work, and is it different from high-speed AFM compared to regular AFM?
Okay, wait, let me go to the picture. Okay, so the basic foundation for both of them is based on the piezoelectric. And based on what I know, I think it is relatively similar for both of them. I think the main difference for the high-speed AFM is the thinner cantilever, which helps them just image the sample at like a higher rate because you have to capture a lot of images in a really short amount of time with high-speed AFM.
Great presentation. Can you go back to the porphyrin experiment? Because I'm trying to wrap my head around the experiment here. In a way, in a sense that the experiment is, how do you measure, how do you make sure that actually touching the sample when doing the measurement doesn't interfere with the actual dynamic of the polymerization process?
Well, in this image, in this image, it shows that it did interfere with it. But I think, like one group said before, you can kind of alter the force that you use to minimize the effect as much as possible. But I believe it, in this image, it does sometimes interfere with your results.
Do they comment in the paper at all about that number that they came up with of 12.4 nanometers per second? Do they?
Yes, they did. They said that they just basically said that they got an average of 35 porphyrin molecules added per second to the nanofiber, which comes out to about 12.4 nanometers per second being added to the nanofiber in terms of its elongation.
Thank you.
Dr. Rita Blake.
Can you go back to figure 16A? So I think you had that in slide. Yeah. So I'm trying to understand more with A. So from 0 seconds to 13.5 seconds, can you explain what the down arrow, there's a white down arrow, what that's indicating?
I believe the down arrow is just the motion of the actin, but I'm not sure. I don't think it directly addresses that in the paper at all that I read.
Because it looks like there are, when it's broken, that the two halves are doing two kind of different things, and I believe that's what the paper might be trying to indicate.
I think it's indicating the direction they're moving and how they're behaving differently with the pressure added, but I'm not exactly sure.
So, but moving in what way? Is the whole polymer itself moving?
The separate ends are moving differently once they apply pressure. Yeah, they're growing in different ways.
So wait, so are they moving or are they growing?
I'm not sure.
Okay.
All right, last question.
So I think we talked about, nothing in life is free. And so you want speed and you lose resolution. And I'm wondering if you could just maybe comment on why that is, what the difference with AFM is. We talked a little bit about this yesterday. What makes going fast challenging in this case? We're not using a camera, right? We're scanning a finger. What makes that difficult to do if you can move it as fast as you want?
I'll try to answer that question. When we go at fast speeds, sometimes it's hard to scan the exact sample because the tip has to move up and down depending on the topography. So it could reach a bump and skip up and miss certain gaps and edges, and it won't perfectly measure the perfect a very smooth surface. You might not be able to get every little impurity. When you're trying to go very quickly, it might skip over some stuff.
We also talked to researchers yesterday who said that in a lot of cases they use the high-speed AFM for like surveying data, like in the beginning of their process, because it's like a little bit or like a lot less clear than when they're using the AFM. But if you just like want to get an image like at a really fast pace so that you can like begin your research or dive deeper, like this is a process that you'd want to use.
Great, thank you.
Thank you.
Okay, and last but not least, group 6. We are now moving away from AFMs and onto optical microscopy.
Can you
Wait, wait, wait.
No, that one's. Oh no, because mine's different.
No, can you pull it up?
Mine's different.
No,
Just
like
bring your
that.
computer. Okay, hi guys. This is our presentation about super resolution optical and confocal laser microscopy. I'm Ethan.
I'm Yuji.
I'm at Yichen Lin.
I'm Victoria.
I'm Lin Chen.
So this is what we're going to go over in our presentation. Yeah.
Well, you guys can read.
Oh, my bad.
First of all, we will talk about the super resolution of the microscope and the confocal laser light microscope in other image, nanoparticle and dynamics. There are our keywords. Light microscope and
Go.
confocal laser scanning microscope, CLSM, and stimulates emission detection, STED.
Okay, so basic optical microscopy has provided the foundation for advanced techniques like confocal laser scanning microscopy, which stands for CLSM, and recently super resolution stimulated emission depletion, which stands for STED, microscopy, and stochastic optical reconstruction microscopy, which is STORM. These newer methods are powerful because they give us 3 big advantages. First, they are in situ and non-destructive. That means we can look at all samples, like cells or tissues, as they naturally are, without harming them. We can even watch tiny particles, like nanoparticles, move inside living tissue. Second, confocal microscopy uses a focused laser beam to scan the sample 1 point at a time, and there's like kind of like a diagram to
It's a
represent that.
pinhole right there. That's what makes the microscope confocal, is a pinhole to focus light.
Yeah. So a special part called a pinhole filters out blurry light from areas that are out of focus. This gives us much clearer images and let us take thin, flat slices of a sample from top to bottom along the z-axis. We can then stack these slices to build a 3D image, helping us see where things are inside cells or tissues. So here is a schematic showing how the laser scans and the pinhole filters for this effect. The inset also compares the improved resolution of standard wide field microscopy to confocal and then super resolution techniques. We finally advanced CLSM into the super resolution realm using techniques such as STED and STORM and bypass the diffraction limit inherent to light microscopy of typically around 200 nanometers. STED improves resolution in the X, Y, and Z directions by using a second laser shaped like a donut, which can be represented above there, which turns off unwanted light around the edges. This makes the glowing spots smaller and more precise. STORM works differently. It uses special blinking dyes, so only a few tiny spots glow at a time. After taking thousands of images, we can pinpoint where each molecule is and build a super detailed picture. These features working with live samples, showing movement in real time, making 3D images using fluorescent tags, and zooming past normal limits help scientists study how nanoparticles, like exosomes, behave in complex systems like the human body. That's why these methods are now widely used.
Okay. In traditional light microscopy, image contrast arises mainly from the absorption, scattering, and reflection of light by different sample components. While useful, these methods have limited specificity and resolution. Instead, confocal and super resolution methods heavily rely on fluorescence. This means we tag molecules with special fluorescent dyes called fluorophores. These dyes absorb one color of light and emit another, which we can detect. The difference between the light we shine and the light that comes out is called the Stokes shift, which can be represented from that picture above. This shift lets us use optical filters to block out the laser light and see only the glowing parts we care about. To highlight specific molecules or nanoparticles, we use fluorescent stains that stick only to our targets. So when the laser hits, only those parts light up, making it easier to see exactly what we're looking for. Normally, the resolution of a microscope is limited by the wavelength of light, which means you can't see anything smaller than about 200 nanometers. But most nanoparticles are much smaller than that. So that's where super resolution methods come in. Instead, microscopy, we use two lasers. One excites the sample, and the donut-shaped laser turns off light around the edges. This makes the glowing spot tiny and sharp, helping us see in all three dimensions. In STORM, we use dyes and blink randomly. At any moment, only a few molecules light up. By taking thousands and thousands of pictures and recording the exact spots that light up each time, we can build a very detailed image with nanometer precision. And this table is shown below. It summarizes the key differences between the methods in terms of image contrast, resolution limits, basic operating principles, and whether they provide three-dimensional imaging. For example, you can see that traditional microscopy has limited resolution and no 3D imaging, whereas CLSM improves resolution and offers optical sectioning for 3D images. STED and STORM go even further, achieving super resolution and higher precision molecular localization. So by understanding and controlling how light is absorbed, emitted, and manipulated, these advanced microscopy methods let us see the nanoscale world inside living samples in 3D with unprecedented clarity.
Okay, so as Yuji explained, but we also could have inferred from the name, light microscopy uses light to visualize small samples under the microscope. But it is a double-edged sword since light limits the resolution of said samples. That's because of a thing called diffraction, which means when light moves through a narrow aperture or around edges, it spreads out. This causes a blurred view of the sample and the inability to distinguish two closely spaced objects. And the diffraction limit that was proposed by Ernst Abbe is that the smallest resolvable distance between 2 points using a conventional microscope may never be smaller than half the wavelength of the imaging light. So if we move our eyeballs to the first picture at A, we can see that the light goes through the aperture. The center, the very bright circular center, is called a airy disk, and there's fuzzy light around the edges. And moving to B, you can see two apertures together. You can still tell that they are two different points, but the center is seemingly together. And if we move to C, you can't really separate the 2 points anymore because they are so close together and the fuzzy light blurs the edges. Oh wait, move back, I forgot the second picture, sorry. So the light is moving in a straight line, but once it hits this metal rod, it spreads out afterwards when it goes through.
Now let's talk about how lasers and filters can help increase image contrast. Lasers emit coherent light, which means that their wavelengths all have the same frequency and constant phase difference. So meaning the peaks and troughs of the waves align consistently. So it looks like that, the top image right there. So this coherence helps reduce background noise and significantly enhances the clarity of the image of the photo you took. Lasers can also target specific wavelengths to excite fluorescent probes. this selective excitation helps to minimize the influence of non-target areas. So areas that you don't want to excite just yet will not show. Then next, let's talk about filters. Excitation filters come in different types: high pass, short pass, and band pass. A band pass is the combination of high pass and short pass, which, unlike, like the name, high pass blocks low frequencies. And low pass filters which block high frequencies. The frequencies between the cutoff points of the two filters form the pass band. By ensuring only the appropriate wavelengths pass through the filters, excitation filters in the end create clearer and more accurate images and increase the fluorescence signal, and thereby improving the signal to noise ratio. Dichrotic mirrors, or dichrotic filter, as said over there, help direct specific wavelengths to while allowing the emitted fluorescence to pass through, which selectively reroutes and increase image clarity.
Okay, there are 4 different techniques of the CLSM. First, the traditional one is the single photon CLSM. Its principle is using single wavelengths, excited fluorescence, and the pinhole filters are out of the focal light, and it has some advantage and disadvantage. The benefit is high resolution, about 200 nanometer, and the optical selectioning capability. The drawback are high phototoxicity and limited penetration. Depth is about less than 100 micrometer. Second one is the multi-photon microscope, also called MPM. The principle is using long wavelength laser excitation and relying on 2 or 3 photon absorption. These advantages are deep tissue imaging that can reduce the photo damage and the low spontaneous fluorescence. Background that not require pinhole, and the disadvantage is apparatus is expensive and the resolution is low, about 300 micrometer. The third one is FLM, fluorescence lifetime imaging microscope. Its principle is detect fluorescence lifespan. Not only intensity, it can distinguish nanoparticle microenvironments that can using test pH scale and temperature sensitive probes. Last one is spectral CLSM. The principle is spectral detection of emission spectra to distinguish overlapping fluorescence signals. It can be applied in synchronous imaging of multi-color labeled nanoparticles.
Fluorescence spectroscopy is a type of electromagnetic spectroscopy that analyzes fluorescence from a sample. This technique uses different fluorescent labels to allow nanoparticles to emit light at different distinguishable wavelengths, which allow differentiation. Absorption spectroscopy is spectroscopy that involves measuring the absorption of electromagnetic radiation due to its interaction with the sample. Different nanoparticles can be differentiated based on their unique absorption characteristics. Each type absorbs light at different specific wavelengths, which can be measured to find out what kind of nanoparticle it is. DLS, dynamic light scattering, is used to determine the size distribution of small particles in suspension. This method is really good at differentiating nanoparticles based on size, as I said. Next, we have the Raman spectroscopy, which provides information about molecular vibrations, which can be indicative of the particle's chemical composition. And it relies on releasing inelastic scattering of photons, which is also known as Raman scattering.
Okay, so I have a lot of stuff to explain and not that much time, so I'm just gonna go quickly. You guys have know what resolution is. I think they've explained it a bunch of times, but basically the minimum distance where two objects are identifiable as individual entities. So this is a pretty complex table, but sorry, my script's like messing up. Okay. So stuff that isn't as important is like the acquisition speed. That's like how long it takes to gather a sample. Imaging depth. That's just like how deep you can go into a sample and then still like get a good image. Light intensity is self-explanatory, and then there's disadvantages to each. So I'm just going to be explaining like the different types of microscopes that we use like for our topic. So the conventional confocal microscope, it's pretty foundational. It uses a pinhole like we explained earlier. And then the reason, this is like a really, really good confocal microscope because this is pretty much like as low as high as the resolution gets with normal light because of the diffraction limit, which we explained earlier. And the reason it's high, there's more resolution on the XY is because when the light is coming to focus on the sample, it focuses like if the screen is our sample, like the light comes in and focuses, but like the Z axis is the front to back, and it's only focused on one plane here, so that's why the resolution is lower. And then the rest of them are super resolution. So super resolution is just like microscopes that are able to overcome the diffraction limit of a normal optical microscope. So SIM stands for structural illuminated microscope, and it basically like creates an illumination pattern over the sample, and you have to use a computer in order to create the image because it uses a lot of math and stuff about that because you can't like it goes into like high resolution than you can see with just normal photons. And then STED is with the laser, and then you use it to you focus it using the like donut, and then the 2D has high resolution on the XY plane, and then the 3D has high resolution on the Z plane, so front to back, but you sacrifice some XY resolution for that. And then STORM is the one where you
select the individual molecules to, or SMLM, but that includes Storm, and she was talking about that earlier, where you like choose individual molecules and you create a higher resolution image with that. These are just some images with conventional light microscope compared to SIM, so you can see it's like double the resolution. So you can see 10,000 nanometers with CLSM and then 5,000 with SIM. This is just TEM on the left side to show you what it actually looks like, and then the comparison between LCSM and STED, and then you can see STED's a lot more focused. And then this is SMLM, where it takes individual parts. So you see it takes a green and pink part, and then it shapes it together. And then there's also 3D reconstruction. And then these are nanoparticles. You see the green on the left side is protein-based, and the left side, the red is the lipid-based, and then you can see them layered. And then this is, the graph is just something like the color, like in relation to the length. Real-world applications, drug research, so you can see how drugs like affect individual cells, viral infection, like you see how viruses work and when they're like going into other cells, intracellular interactions, how cells communicate with one another, and then intracellular, so how a cell communicates with itself, and then 3D reconstruction, so like I was talking about the z-axis or front back, you just take a bunch of photos and then smash it together and make a 3D.
Okay, so in conclusion, CLSM, though not a super resolution technique itself, provides a foundation for implementing/integrating techniques with super resolution techniques that have achieved resolutions that go beyond the diffraction limit that was first proposed by Ernst Abbe. Having techniques such as STED, stimulated emission depletion microscopy, ISM, image scanning microscopy, and SIM, structured illumination microscopy. By enabling high precision imaging of nanoparticles, we can now dive deeper into the structures and processes of various organisms as this technique continues to evolve and unlocks more potential for improved, detailed imaging in the nanoscale.
Wait, no, we still have questions.
Okay, Jordan.
Hi guys, thank you. That was a great presentation. So just to clarify, I wanted to make sure. So these colors that you're showing in all these images, this is actually means something, right? Like we have like it's actually quantitative because this is what we see with our eyeballs, right? This is photons within this visible spectrum. I just wanted to clarify. That's what's happening, right?
Oh, okay, so yeah, sorry, I just had to skip through.
You can show that last image if you want. So this one.
Oh, this one? Okay. So this one you could actually see in real time. This was in Dr. Bentalila's lab. So this is when we're singling out here. I'll describe each one. So the green is the it's part of the cytoskeleton. I forget the exact name again, but it's like microtubules or something.
That's okay. So it's emitting green photons?
Yeah. So basically, it's marked with GFP. So green fluorescent protein. So you, in order to activate it, you have to emit shine a lower wavelength. So we shined a blue laser on it actually to activate the green fluorescent protein. And then so for each of the other ones, we did like really deep blue light for the blue, and then like I think yellow laser for the red. And then these are all individual, and then the last one is all of them smashed together.
Okay, great. Thank you.
So the colors that were shown here are not inherent to the cells, right? So how do we get these colors and see these different portions of the cells?
So basically, they're fluorophores, which are like they come from natural like organisms. What? Yeah, they're dyes, but they're generated synthetically, so that we don't have to like kill a jellyfish every time we want like GFP or something. And then we create them so that they're only in a specific part of the cell, like the blue is the nucleus right there with the BFP. And then we can see that by shining the laser on it. So we're singling out only the blue color and only the nucleus with that.
Hi.
All right, so if the sample was really thick, I know that the focus of this paper was on really tiny things, but what kind of microscope would be better for something that's thicker instead of super res? Or how does the confocal handle a really thick sample?
So you can see that the imaging depth is actually very thin for all of these. It's actually very thin for the SMLM one, but I think that I'm not actually sure, but if I had to guess, I would think that the conventional confocal one would be best at getting the deepest images. But as you can see, there's all really thin samples. Like actually the electron microscope even gets is it 100 nanometer sample or no? Oh wait, this is micrometer. Oh. So then it's pretty thick sample, no?
Yeah, so
It's really thick sample actually. At the beginning. Did one of you guys put that? That's yours. What? One more? Oh. Sorry. This one's yours.
Yeah, so it's just the main point being if you when you start sectioning really tiny, right, when you have the super resolution, if your sample's really big, it would take you hours and hours and hours, and then your thing might be dead by the time you finish imaging it, right? So there's always this kind of trade-off between resolution and time. How much time do you have, and how much resolution do you need? But yeah.
All right. Do you want to explain that to her, or Oh, wait, did we answer your question, or
You're good.
Okay.
You're good.
Sorry.
Well, you have it here. It's multiphoton microscopy. If you remember our class, we explained multiphoton microscopy by changing the color of light, right?
Yeah.
If you if you go further into the red and you shine that red light through your skin, for example, right? You're gonna have less scattering, and you're gonna see red light going through your skin. If you take visible light, right, like green light, it will barely penetrate a few layers of your skin. So in multiphoton microscopy, this is what we use. We use red-shifted light toward the infrared, and doing so, we can go much deeper. Okay. If I have time to ask you maybe one great presentation, by the way, thank you so much. Actually, you brought a lot of things that we even didn't cover, so kudos to you guys. Can you maybe explain to the audience really what STED microscopy is and how it is implemented? How can we turn a regular optical microscope, which is very good, to something that can measure even smaller things? What is the trick, essentially, in STED microscopy that we implemented?
Okay, so I think that I might have rushed past it, but essentially it uses this super powerful laser. I think it might be we might have a picture of it on the slides, maybe perhaps on the first slide.
Right there.
No, I'm talking about the actual machine. Yeah, this is a STED microscope. So there's a box to the right that generates this super powerful laser, and obviously it has to be this is a very well, all of them are, but this is a very, very precise machine. And the laser is let's go back to the donut. Okay, this takes a really long time actually. So this is the donut that it creates to focus on the sample. So can you explain this one better, because this is yours?
Okay, so it depletes fluorescence around the edge of the excitation spot, so it's effective in shrinking the emitting area and improving the spatial resolution in all 3 dimensions.
And it's also it can be very damaging, I guess, if you're trying to do biological samples and stuff because of the laser. So and then also I think you need very strong fluorophores or dyes that won't get broken down by the power of the laser.
Thank you.
All right. Thank you, guys.
Where is it? Thank you slide now.
Go more. Go more. Go more.
Good.
Go more.
Thank you.
Alrighty. Okay, so I have some closing remarks and then we get to eat food. Alright, so we just want to let all of our students know that we want to help you succeed even beyond this course. So we had you for 5 days, but you are our students forever. And so actually at the beginning of this course, or on Wednesday of this week actually, we had a student come by in the morning, and he was a student in our nanoscience lab class 2 years ago. Now he is a UCLA undergraduate student, and he is working in Professor Rick Kainer's lab in the chemistry department. And he came by and he told us that he used his experience in our summer program and wrote about that in his college essays. And he thinks that some of the things that he learned in our nanoscience lab class helped him get a position, a research position in Rick Kainer's lab, because Rick Kainer is also a nanoscientist and uses some of the same things that we talked about in these classes. So please stay in touch with us. It was a very rewarding experience for me to hear from this student. And so if you're going to college or university, getting a PhD, MD, starting a business, we want to know about it. I also wanted to tell you guys about our CNSI Nanovation competition, which applications are open in December and January, and this is a competition that combines nanoscience and entrepreneurship, another great experience for students to apply things that they learned. And then we have other CNSI summer sessions courses. So you guys are in nanoscale microscopy, but we have an applications of nanoscience course, which combines nanoscience and entrepreneurship, and nanoscience lab, where you do a series of nanoscience experiments in a laboratory. And so we write letters of recommendation, mostly me, if I'm being honest. So you can email me, and I will help you get that set up. So we can't specifically help you get into UCLA. That's not a thing. The UCs actually don't even accept letters of recommendation, okay? But we will write letters of recommendation for other universities that you're applying to, other summer programs that you're applying to. We are here to help, so keep us in mind. And then one of our learning objectives was about reflection and introspection. So I'd like all of our students to take a moment to think about what you did know about nanoscience and microscopy before you started this course, and think about what you learned over the last week. So about science, about microscopy, about working with other people, and a little bit about yourself, right? So hopefully some of you are interested in being scientists after taking this class. So while you all are thinking about that, I'm going to see if our instructors have any closing remarks that they would like to say. Pause.
While they're all thinking. So, Shailene did a great job of introducing everybody. My name's Adam Stieg. Parents, thank you for sending this brilliant crop of young minds to spend a week with us today. I'm one of the associate directors of the CNSI. I work and oversee the technology centers with Laurent and Hong and Matt and the folks that have helped the students navigate basically a fire hose of information that they've been exposed to for this entire week. It's always great to see new crops of young students coming in here asking tons of questions. As I said to them, I think yesterday and when I did an intro on Monday, what we want is for you to leave here with more questions than you came in with, right? To be excited and inspired to go out into the world when you go back, and wherever it is that you go, to keep asking questions. Inquiry is the sort of, founding principle for discovery, and we need a lot of you all to solve some of the challenges that we all face for our futures. And so thank you to the parents for sending them here. I know many of them, this is the first time they may have gone away to a university and be in a dorm and do these types of things. I hope you all learned something. I learned apparently yesterday not to use the numbers 6 and 7 in a sequence together. So we learned from each other over the week. It was a really fun time to spend time with them. I'll speak on behalf of Laurent and Haley and Matt and the rest of the team. they've said the same things. And the presentations are incredibly impressive. You guys digested a huge amount of content. Really appreciate the effort and the hard work that you put in. And I'll just end and say, Shailene never gets the big thanks that she deserves, but Shailene is the glue that holds. She's the one that holds this whole enterprise together and keeps the ship moving. And so thank you, Shailene, for all the hard work. And with that, I guess we'll probably just wrap. I think we have food or a gathering afterwards. I know that most of us will be around. Rita is raising her hand like a very good student.
Right.
But we'll be all around to answer questions. It would be great, I think, also to a
Group
picture,
photo.
group
Yep,
photo, and yeah, we're gonna do a group photo. So before everybody runs off, right, let's try to get a couple pictures. And again, just to reiterate with Shailene, please remember us. We'll remember you. You know where to find us, and anything we can do to help you on your journey, wherever it takes you, we're happy to help. So thanks so much.
Yeah. So thank you, everybody.